Good evening. I'm Craig Thompson, President and CEO of Memorial Sloan Kettering Cancer Center. I'm pleased to welcome you this year to the 2020 now virtual symposium called Major Trends in Modern Cancer Research. This is the 15th year we've held this symposium where we try and bring you up to date on the latest information of the scientific research that's carried out here at Memorial Sloan Kettering to understand cancer in all of its manifestations. You might wonder this year, while we're having this symposium, when the world is being consumed in the COVID-19 pandemic. Well, first, cancer is a predisposing condition to the risks of COVID-19. And we take care of at any one time here at Memorial Sloan Kettering 150,000 patients undergoing active cancer treatment. In doing that, we had to adjust our research mission to understand, because cancer patients are more susceptible to immunologic diseases and infectious diseases, and to provide the best understanding to the world as it looks out at us of what might be the best possible treatments of cancer during this period where patients could be potentially exposed to COVID-19, which itself is a life-threatening disease. This year, since March, when New York had its first case of COVID-19, our researchers have produced over 201 research articles informing the world of the best possible practices of treatment of both COVID-19 in cancer patients and of the treatment of cancer in patients that might otherwise be exposed to COVID-19. And you'll hear a little bit about this today at this symposium, but equally important why we're having this symposium is that cancer does not shelter in place during even a pandemic like COVID-19. This year, worldwide, 5 times more patients will die of cancer than will die of COVID-19. And so this is a devastating illness that can affect all tissues of our body that self-renew. And so the understanding of cancer and improving its diagnosis and treatment is fundamental to world health. And so we really like to participate in this symposium to get you, the students who are interested in cancer research, to understanding some of the latest cutting-edge research and the tools that we can bring to bear on understanding this disease, cancer, and to get you excited about how it is that important research is undertaken, specifically in cancer, but to understand all of human biology. Tonight, we've invited 3 of MSK's investigators who specialize in different areas of cancer research to present short talks on what they do. We think their talks will help illustrate how exciting and rewarding scientific careers in cancer research can be. And they even may inspire some of you who are listening here tonight to think about pursuing careers in this area. Before they get started, I want to tell you a little bit about how we've organized this virtual meeting. It's a little different than in the past. Each presentation will be 15 minutes. And we'll have about 10 minutes after each speaker speaks to have questions and answers. We're going to keep your cameras and microphones off for this event. So if you want to ask a question, please ask whatever questions you find relevant to your understanding of what they're talking about or cancer in general. You ask your questions in the chat section. Our Dean of our graduate school, Michael Overholzer, is here and in the audience. He will be looking constantly for your questions in the chat box, and he will share them with our speakers to keep the flow going as each of the speakers completes and answers questions. Each year we have gotten in the past, when we've done it in person, really great questions from the audience that have not only stimulated all of us to think about the areas that are being talked about more deeply, it actually gives us new ideas in terms of what we should be pursuing in the laboratories. In the past, I've given out an award to the first student who stumps one of our speakers to get them to say, "I don't know," because that's an area where cancer research has to be double its pace to really understand this problem that we're facing. We'll try and figure out how that goes. We'll ask Michael to identify and announce whenever that has actually been achieved tonight. If there's time at the end, we will also bring all of the speakers back. You'll see they're masked right now and up front, but we will bring them all back to ask any questions that wrap up the whole session if there's time during our period. And finally, I want to remind everybody that if you like what you hear tonight, you can follow us on social media so you can stay up to date on the research that's going out throughout Memorial Sloan Kettering, as well as some of the future events and exciting internship programs that we have here. So with that as the introduction, I want to now introduce our first speaker, Karuna Ganesh. Karuna is a medical oncologist who specializes in the treatment of people that have cancers of the GI or the gastrointestinal tract. She is an assistant member also in our research program in the molecular pharmacology program of the Sloan Kettering Institute. Karuna grew up in India. She did her undergraduate work at the University of Cambridge in England, and she pursued an MD, PhD degrees there as well. We were lucky she crossed the Atlantic and now is a member of our faculty. In her laboratory, which she has just started in the last couple of years, she explores the use of balls of cancer cells that we call organoids. This is a new technique that's been developed, she'll tell you about, to allow to understand how cancer cells cooperate in 3 dimensions to gain the ability to metastasize and cause harm to the body as they grow and spread. So with that introduction, I'm going to ask Karuna to come up. I'm going to go get masked and sit down, and I look forward, I hope as much as you, to her presentation. Karuna.
I'm very excited to be here today to tell you a little bit about our work. As Craig mentioned, I'm a physician scientist, and as a medical doctor, I take care of patients with advanced cancers. And I want to start off this session today by telling you a little bit about one of my patients. And I think this will give you a sense of why my lab does the science that we do in the hope of finding better treatments for patients like this one. This is the story of GH. He was actually one of the first patients I encountered and treated here at Sloan Kettering. She was, at the time, a 28-year-old woman. And just a few weeks before I met her, she had actually been in the throes of labor, actually giving birth to her first child. And in the course of labor, the baby actually got stuck. And while the obstetrician was trying to figure out what was going on, they felt around and they found what they thought was a mass, a lump. And they weren't sure what that was, but she eventually had a C-section. The baby was fine. And then she went back to get this MRI that you're seeing here. And what they saw, unfortunately, on this MRI is this large area that occludes, really blocks the entire length of her rectum, the last part of the intestine before it comes out to the outside world. And this was, in fact, a stage 3 rectal cancer, diagnosed as she was in the middle of labor back in 2012. We treated GH, as we often do for patients with this disease, with 6 months of treatment. It's pretty intensive treatment. She got chemotherapy for 4 months, then radiation together with chemotherapy to the rectum and the pelvis for 2 months, and then eventually she had a major surgery to cut out this entire tumor, the lymph nodes, and with the hope, of course, of curing her. We hoped that the cancer had gone away after all this intensive treatment. And indeed, the cancer did stay away for about 2 years. But unfortunately, after that, back in 2015, what we did was to do this PET scan. And what you're seeing here lighting up in the middle of her lungs was a new tumor. And this was, when we biopsied it, exactly the same tumor that she'd had back in 2012, which we thought, we all hoped desperately that we had cured, that we had gotten rid of. But unfortunately, we had not, because even in 2012, there were probably microscopic seeds that had left this rectal cancer and spread to the lungs. And given enough time, in this case about 2 years, this tumor started to grow as what's called a lung metastasis. And when we looked further, we found, unfortunately, in her brain a large mass, a brain metastasis. Again, a biopsy confirmed that this had come originally from her rectum. We treated her with all of our best, most aggressive treatments at that time. She received, over the course of the next year or so, surgery to remove these tumors in the lung, to remove the tumor in the brain. She was also treated with 3 different types of chemotherapy, 2 different types of what we call targeted therapy, treatments that are targeted specifically to specific mutations or alterations in the cancer cells. And then also, she was actually one of our first patients on a clinical trial with immunotherapy, training the body's own immune system to attack these cancers. And she did well for a while, but unfortunately, in less than 2 years, just over a year after we first found that first metastasis, she now had much more extensive, much more aggressive disease in her lungs. And she rapidly developed new brain metastases. And unfortunately, by April of 2016, she had died. And this was, of course, a very potent and tragic case in this very young lady. But this is unfortunately the reality for most of our patients who have metastatic disease that has spread from the site of a primary tumor, the place where the cancer begins, to a different organ in the body. And indeed, we know now that metastatic cancer is typically incurable. In fact, metastasis is what causes 90% of cancer death. We now know that actually, as we learned with GH, the process of metastasis, the process of the spreading, starts very early in the course of a tumor. Almost as soon as a tumor has started to grow in an initial organ, whether it's a breast or the lung or the colon, these microscopic cells from the tumor can start to spread to different organs. And they can stay there throughout diagnosis, throughout treatment, as they did with GH, as metastasis stem cells, cells that are dormant, not growing, hiding out, but that have the potential, given time, to grow into these macrometastases, which is what we can see on scans like we just saw. And once we have macrometastases, we can treat these patients with chemotherapy, targeted therapy, immunotherapy, but all we can achieve is to debulk these tumors, meaning that we get rid of the bulk of the cancer cells, but we're unable to get rid of them entirely. We're unable to get rid of these stem cells, the metastasis stem cells, that with each round of treatment get more and more aggressive, grow back bigger and stronger until they eventually take over the functions of normal, healthy organs and kill people. And indeed, it is these metastasis stem cells that drive the process of these tumors spreading and growing in different organs and eventually kill people. So we have, of course, been very interested to understand what makes these metastasis stem cells tick. And what we've now learned is that the key feature of metastasis stem cells is that they're able to constantly change their behavior. Genetically, they're exactly the same. They have the same mutations, the same alterations in these cells that were there in the primary tumor often. But what they have is this ability to turn on and off different programs of genes which allow them to change their behavior, to adapt to surviving outside the breast or the lung or the colon where they first started, to adapt to surviving in the circulation while they're spreading to different organs, to evade the immune system that's trying to kill these hostile foreign cells in different organs, and eventually to grow, to set up shop in a distant organ and resist therapy. And it is this ability to change, a feature that we call plasticity, that makes metastatic cancers so lethal. And this has been something that we've been trying really hard to study. And this has historically been very challenging, because ideally what we want is to be able to take these cancers out of patients and to be able to grow them in three-dimensional conditions, in conditions that allow us to watch and understand how these cancer cells change and adapt to different stresses, such as different treatments or different environments. And it has historically been really hard to be able to generate model systems in which we can study cancer from people directly in that way. And all of this changed about a decade ago with the invention of a new model system, which is called the organoid. And I'm going to take you now into my lab to tell you a little bit more about what organoids are. Welcome to my lab. So here we take tumors that have been cut out of patients undergoing surgery at our hospital, and we convert them into little three-dimensional mini tumors that we call organoids that we can use for research. This is Ahmed, a graduate student in my lab. Ahmed, can you tell us what you've been doing?
So last week, one of your patients had surgery to take out one of his liver tumors. And what I've done is I have broken down that tumor into tiny clusters of cells, and I've been growing them into organoids. And right now I'm just checking to see if they have grown into organoids.
Well, let's take a look.
Absolutely.
Aha.
They've grown very nicely into organoids.
Yeah, you can see lots of different sizes and shapes. Let's zoom in and take a closer look. Wow. As you can see here, you're seeing these three-dimensional structures, and this is, of course, exactly what tumors are in patients. They are three-dimensional, and what you can really nicely see is that these tumor cells that grew from the colon, from the intestine, have formed a little tube-like structure. And so these are sometimes called mini guts because you can see the tube of the gut. What's really powerful about the organoids is that firstly, we can grow them from lots and lots of different people with relative ease. Over 80% of patient biopsies, even tiny little biopsies, can be grown very, very efficiently and quickly within a period of days to weeks, as Ahmed has done here, into these organoids, which we can then use to test lots and lots of drugs. And what's really powerful about them is, as you can see here in an organoid from a different patient, we can see that these organoids, just like real tumors, consist of lots and lots of different types of cells, and they change over time as they grow, as they regenerate, as they respond to treatment. But often in people, we know how to kill the green cells, the rapidly dividing ones, with chemotherapy and targeted therapy, but we don't know how to kill the cells that are not growing fast, and that's been really hard to study, these dormant cells. But what Ahmed has done here is develop a way to label in the organoid these dormant cells with this pink marker, and we now have a way to study these dormant, slow-growing cells that can sometimes wake up and kill people, and we can figure out how they do that and how to attack them. And this is what makes organoids really powerful. So now that we have this powerful model system, the organoid, what can we do with them? The first question we had was, what is the extent to which the organoid captures the behavior of cancers in people? And perhaps most importantly, to what extent can an organoid help us predict how a patient's tumor is going to respond to treatment? So we had a way to do this experiment. And what we did was to take patients like GH who have rectal cancer, and we were able to get tiny biopsies from their tumors before they started any treatment and make organoids, as you can see here, from those tiny little biopsies, which are less than a millimeter cubed, and expand them. And we could also take these organoids now and transplant them back into the rectum of a mouse. And so now we could test in parallel how the organoids respond to treatment and also how the mice respond to treatment. And at the same time, the patient is getting those 6 months of chemotherapy and radiation that I told you about. And so we were able to test in parallel the patient's response to drug and the organoid response to drug. And what you're seeing in the bottom is that actually the organoids represent what's happening in people really well. What you're seeing in this panel is a patient whose colonoscopy, which is what you're seeing here, shows a nice clean tube of the gut. There's no tumor left behind after treatment. And similarly, the mouse too has a nice clean tube that you can see through. No tumor left after treatment. On the other hand, this other patient, despite all this treatment, continued to have this big, bulky, ugly tumor that's blocking the inside of the gut. And the mouse too continued to have a big, bulky tumor. With this now, we know that the organoids reproduce patient-specific responses to drug, and now we can start to develop prospective clinical trials looking forward to see if organoids can now predict patients' responses of drug that we can then use to test in patients who may not have any other good options. We can, of course, also use organoids to help us understand the basic elements of metastasis. And what you're seeing here is a cross-section of a colorectal cancer tumor in a patient. And these green cells are the ones that begin the tumor. These are the tumor-initiating cells. But as they start to invade and spread to distant organs, as they start to metastasize, these cells have lost this green marker. And instead, they've switched into this red state, which is a metastasis stem cell state. And they've turned on the production of this red molecule, which is something called L1-CAM. And the reason we know that this switch happens is because now what we could do is to take metastases directly from patients, separate them into the red cells and the green cells, the L1-CAM high cells and the low cells, and ask which ones are better at making organoids and tumors. And we found that across all the different types of patients we saw, it was always the red L1-CAM high cells that were able to make new organoids, that were able to make new tumors in mice. And so with this information, we now learned that all of these now labeled in brown L1-CAM producing cells are able to grow into organoids that contain both brown and non-brown cells. So every tumor or every organoid starts off from a brown L1-CAM cell. And now what we can do is to take this forward towards making drugs that block the production of L1-CAM and prevent these cells from growing into organoids. And that's something that we are doing here. Finally, I want to remind you that tumors don't grow in isolation. Cancer cells don't grow on their own. These cancer cells grow in a microenvironment that includes immune cells, that includes blood cells, that includes connective tissue producing cells. And we have to really understand the interactions of the cancer cells with these other cells. And this is what we call systems-level biology. And this is something that we are now able to do in a patient-specific manner with these organoid models. We can take cancer cells and grow them into these yellow cancer organoids. We can also get blood from individual patients, from the same patient from whom we made an organoid, and get the immune cells from that blood and add them to this mixture with the organoids. And of course, when we're thinking about colorectal cancers, they're growing inside the gut. And our gut is full of bacteria, some of which are good for the cancer, some of which are bad. And now we can get stool from these patients, get the bacteria out of the stool, and put them inside the tube of the organoid and understand how these different components interact with each other. And what we can do now is to actually take videos and visualize how the pink immune cells, in this case, are killing the yellow cancer cells in the organoids, and understands what's different about the cells that are killed well and the cells that survive and can resist immune activation. And similarly, we can take green fluorescent bacteria and put them into the tube of the organoid and try and understand how that works. So putting all this together, what we really are at now is a new era of cancer research, where instead of using cancer cells that are isolated and grown in 2 dimensions on plastic or relying entirely on the mouse, we can use the human as a model system, where we can make organoids, make these organoid immune bacterial co-culture systems from patients in an individualized way and really understand in real time what's happening in an individual patient's tumor. And of course, translate that back into making new drugs, better drugs, and eventually run clinical trials, and eventually, hopefully, improve our outcomes for our cancer patients. So with that, I'll thank the members of my lab, our many collaborators, both scientific and clinical, and of course, everyone who funds our research. And I'm happy to take some questions.
So Karuna, we have a lot of questions coming in, and we're going to start with a few that are more general, then we'll narrow into some specific ones. So to start off, these are great questions. How many cells are in an organoid, and how complex can they be? And I'm going to combine a few questions here. Does each organoid contain a stem cell?
These are all fantastic questions. And the answer is it's complicated. So every organoid can start from a single cell, which is a stem cell. But there may be many different types of these stem cells, and that's something that we're trying to understand now. How many different types of stem cells can make organoids? And then in terms of how many organoids, how many cells there are in an organoid, that changes over time, because we see these organoids growing after treatment. They will kill, the treatments might kill some of the cells, others will survive. And this ability to change is really a key feature of the organoid, which of course reproduces what's happening in patient tumors in real life, and that's what's so exciting about them.
We have numerous questions related to dormancy, and so I'm going to summarize them by asking, what makes the dormant stem cells wake up? And really, what is dormancy? Is it a single cell surviving for a long period of time, or is it dividing at some slow rate?
Again, fantastic questions, and I wish we knew the perfect answers to these questions, because we would have better drugs if we did. So I think the point that you made was great, that there's different types of dormancy. There is something that we think of as cell-autonomous dormancy, where the cancer cell on its own decides to go to sleep. And we and others think that there may be some signaling cues. So for example, that cell may have shut off its growth factor signaling pathway. So even though it may be growing in the presence of lots of factors that are telling it to grow, the cell has turned off the machinery that tells it to wake up and grow. And of course, there may be ways with drugs to be able to overcome that. But there is another type of dormancy, which is called tumor mass dormancy, which is overall the tumor is not growing, but that's not because the individual cells are not growing. The individual cells may be growing all the time, but at the same time, the immune cells are destroying some of those cells. So overall, there's a balance, an equilibrium between the cancer cells and the immune cells. And that's something else that's very interesting that many people around the world are working to try and attack.
Great. Many great questions coming through. Is there a way to mix the tumor organoids with normal cells in a culture system to try to see how they interact? And related to that, can the organoids be used to create personalized patient care?
Yeah. Again, great question. So yes, absolutely. So in addition to making organoids from tumor cells, we can actually make organoids from normal cells as well. And certainly it would be very interesting to mix the two types of cells together and see which one wins in different circumstances and how the two interact with each other. And for the second question about personalized cancer therapies, this is absolutely something that we are working towards. And these are things that have to be very rigorously tested. There may be some drugs for which organoids are particularly good models to predict for an individual patient whether or not that patient will respond to those drugs, as we have shown. And there may be other drugs for which organoids are not so optimal. So these are things that we have to, while we're very excited about the potential here, this is something that has to be really rigorously tested in large clinical trials before we can really take that forward to every patient.
Great. What makes metastatic cancer stem cells different from normal stem cells?
Yeah, that's another great question. So that is something that my lab and our colleagues, we've worked on quite a bit. And we know that the normal stem cells, at least in the intestine, are very much like the cancer stem cells. The tumor-initiating cells are like the stem cells that maintain a normal colon. So every day, our colon is shedding cells, and new cells have to be made, so the stem cells are dividing. But when you damage the intestine, those normal stem cells are not working. It's a completely new population of stem cells, a regenerative stem cell population that gets activated. And metastasis stem cells, we think, behave a lot like that. And the reason for that is because the attachments between the cancer cells are lost during metastasis and during wound healing. And this triggers this very different regenerative program.
I think we have time for one or two more.
Okay.
So I apologize, I can't have every question be read out loud. We have obviously some great scientists in the crowd here. So once a cancer cell emerges, how long does it take to become clinically evident?
Yeah, this varies from cancer to cancer, but on average, there are mathematical models that show this, it takes probably about 10 years to become a full-blown cancer. So, so a lot of my patients will ask me, did I do something last year that may have made this cancer come up? And the answer is no, in almost every case. That these things are, mutations happen by chance. And unfortunately, over time, with chance, as long as there's a mutation that promotes growth, those tumors will form.
And finally, we've had many questions about L1-CAM. So clearly, everyone was paying attention to your talk.
Yep.
Very nice. So can you tell us a little bit about whether normal cells express L1-CAM as well, and how do tumor cells turn on the expression of L1-CAM?
Great questions. So among normal cells, we know L1-CAM is required in the developing brain. So to make neurons, the cells that are in the brain and in the nerves, we need L1-CAM. But even though L1-CAM is present in adult neurons, we don't think it's doing anything hugely important, because there are studies that have shown that if you delete L1-CAM in the mice, in adults, they don't really change very much. But the other question about how do cells turn on L1-CAM, that's something that we're very interested in. We know that there's a whole signaling pathway where the cell can detect when it's been detached from its correct context, where it can detect that it's no longer attached to its friends, as is happening when a cancer cell starts to spread to a different organ. And that activates signaling within the cell to allow L1-CAM to be expressed. And that is some very interesting biology, which may offer some opportunities for new drug development also.
Great. Thank you, Karuna. And thank you, everyone, for all the fantastic questions.
And so although Michael actually did do a great job of conveying your questions, since there were so many, and boil them down to the common ones, I did hear Karuna say twice, we just don't know, particularly around dormancy. And the patients she told us about that went a number of years before their metastasis occurred, that we believe occurred from dormant cells, but we don't know what controls that process. It's something she and others are actively investigating in the laboratory. So those were fantastic questions. Our next speaker of the evening is Justin Perry. He is an assistant member of Sloan Kettering Institute's immunology program. Justin is originally from Alaska. He earned his undergraduate degree from the University of Cincinnati and a master's degree from Fordham University here in New York City. He went on to get a PhD at Washington University in St. Louis, and we were fortunate to have him join us on the faculty several years ago, where he studies the signaling circuits that govern how immune cells identify and clear out excess cells that accumulate in our tissues and our body over time, either as they're damaged when we cut or damage our cells, or because we produce cells all the time and they have to be turned over during the process of regeneration, if you will. So I'm very pleased to ask him to join us at the podium here and just to give us an update on some of the fantastic work he's doing in this area.
Great, thank you, Craig, and I have the enviable position of following up Karuna's beautiful talk. I also want to just quickly thank everyone for coming today. As a father myself, I understand that these are trying times for everyone, and I hope all of you know how proud we are that you all are just doing the best you can and really trying to continue your education during this pandemic. I often feel like I should have Craig give my talk because he often says things much better than we can. But as Craig noted, our lab studies phagocytosis and innate immunity. However, I like to actually say that we study how cells eat other cells and other stuff, and we like to drill down to simple analogies. And so it turns out that for life to exist in every species of biological life, there is a way that those cells have to die. So in the examples of the intestine that Karuna was talking about, in fact, the human intestine turns over every week. So many of those stem cells that are becoming new cells, the old ones have to die and be removed in some sort of way. And it turns out that our bodies have many ways in which cells undergo what we call cell death or programmed cell death. And in fact, we have to have mechanisms and systems in order to get rid of those. And as I noted, it's extremely conserved. And so, for instance, if you look to the roundworm species, what we call C. elegans, you notice that, and as well as in flatworms and planaria, you have cell death and removal of those dead cells is essential for their biology. We see this in zebrafish and other fish species. In this case, I'm showing you in the developing brain of a zebrafish. You see the green dots are actual cells undergoing death and clearance. We see this in the fruit fly, the common household fruit fly, or what we call the Drosophila. And again, this is during development, but this is also undergoing in adulthood for flies. And then we get into, like Craig mentioned, into regenerative species. So in this case, we have the Hydra, or the Synardia, which if you cut in half, the cells on each half of the cut die, but that process facilitates them to grow back together, and you get full regeneration and recoupling of the organism to itself. And this is also true in my favorite animal species, what we call the axolotl, or the Mexican salamander. And so what you're seeing here is a limb regeneration where you completely remove the limb. And when you do that, you get this wave of cell death and clearance of those dead cells that initiates a regenerative process. And those, the axolotl are able to completely reform that limb, including all of the muscle and tissue and neurons and everything that's needed there. And of course, we also get this in mammalian species. Here I'm showing you the paw of a developing mouse. You see here how the skin is, see if the mouse works here. You see how the skin is attached between the digits, and there's cell death occurring during development in between the individual digits of the hand. And then so just as a canonical or classic example of this process going wrong, in humans, you see this if you fail for those cells to die and be removed, you actually see this either complete or incomplete, where you basically are unable to disconnect the digits because of the failure of cells to die and be cleared. And in fact, it turns out that every major organ in the human body, every organ and tissue has cell death and cell clearance processes. It's extremely essential for human health. And if you perturb this process in any one of these organs or tissues, you have catastrophic consequences to the host. So as a classic example, we like to think of, for instance, after, in the mammary gland, I like to give this example. So when pregnancy occurs, you have lactation and breastfeeding, but then after that process, you have what's called mammary gland involution, where you have to clear all of the dead all of the cells that are dying, all of the residual milk, this is essential for that process. But for instance, in cases of neurodegenerative disease, you have massive neuron death, and that's a primary cause of the symptoms that come with neurodegeneration. And it turns out that this process is so pervasive that, in fact, viral infections tend to co-opt these pathways. So in the case of COVID, for instance, we know that coronaviruses recognize the receptors that are also involved in this clearance of dead cell process. And this is true of a lot of the viruses we know. There's such ancient processes that the viruses have evolved in order to take advantage of this. And they're not the only disease that takes advantage of this clearance process. So we're here today talking about cancer, and it turns out that cancer also benefits from this normal cell clearance process, and it uses it to develop and to progress and metastasize, as beautifully illustrated by Karuna. So what does this actually look like? And I think Karuna did a beautiful job of showing how we take observations in the clinic and in patients and in different disease settings, and we work backwards into the lab to try and understand those. But as I'm sure many of you know, in the last couple of years, there's been a lot of attention in what are called immune checkpoint therapies, or basically ways of targeting the immune system for anti-tumor treatments. And a lot of that work stemmed starting from the other way. Can we understand the basic biology of these cells and then learn something about that biology and then apply that to the clinic in these clinical settings? And so this is really what my lab focuses on, is can we understand the basic biology of phagocytosis, or of clearance of dead cells? The word phagocyte literally to be big eater, and this is what this would look like. So here you have what's called a macrophage. This is the professional phagocyte in our body, and we have labeled cancer cells with a reagent that tells us when it's been eaten. It fluoresces bright pink. So this will loop on here, but this entire region is a large macrophage eating tumor cells, dead tumor cells. And you can see here, there's a lot going on in this video. And people like myself and my colleagues get really excited when they see these videos, because we can already start to say and generate lots of interesting questions about what we're seeing. Seeing is believing, seeing is generating hypotheses and testing those hypotheses. And so what you're seeing here is this macrophage taking in that cell. You see this fluorescence come up. This is acidifying and degrading and breaking down that cell. You could see it breaking it into small pieces. So like I said, there's a lot going on. And then after it started to break it up, it moves on and eats another cell. All right, so this is what this looks like in real time. So to me, what I see is a lot of unique biological processes going on, and we want to understand those. This is what we want to understand. And what are the unique steps of dead cell clearance? And so over the last couple decades, a lot of important work has made us realize that there is a series of steps. So starting with what's called the smell, the phagocyte has to find the dead cell and distinguish between live and dead cells. We call those smell signals. Once it finds it, it has to actually physically distinguish between death and life, and we call those taste signals. So it's literally tasting specific things on the surface. That triggers the ingestion of that dead cell. Literally, you're eating those dead cells, and then you have to digest it and break it down. Okay, so we have, you smell them out, you come to where they are, you taste it, ingest it, and digest it. And this is what this looks like in real time, okay? So you basically have a phagocyte finding a dead cell, ignoring its neighboring live cells, and literally bringing that guy in and eating it and breaking, and beginning to break it up. And so these processes, again, we call it the smell, taste, ingestion, and digestion signals. I also like to think of this as these are the detectives of our body. So I like to think of macrophages as our homicide detectives. They have to determine what sort of death is occurring, who's dying, is the cell infected, is it your own cell, or is there something wrong with this cell? So there's a lot of things that they have to do. And in our minds, this is incredibly important to understand, because if you can understand these signals, for instance, if you can understand how a phagocyte, such as a macrophage, eats these dead cells and degrades them, you might be able to make those cells eat more dead cells. So can you make them eat more or eat less of these dead cells? If this process is actually driving the development and progression of tumors, can you find ways of targeting it and playing with this and making those cells do more or less of this process? And for us, an even more exciting option is, can we change the way those phagocytes eat those dead cells? And so what I mean by that is once that the phagocyte has taken up that dead cell, there's all sorts of biological processes that go on inside of the cell. And all of those processes contribute to making this a healthy process. So in our minds, it's can we make these cells do this in an unhealthy way? And if we can do that, maybe we can drive an immune response, for instance, to a tumor or to an infection, and really make our own systems target the cells in a better way. And so just to kind of highlight this, I didn't want to inundate you too much with the specific targets, but this has been a major focus in our lab over the last bit of time. And so what you would see in a normal control cell, this is what that process looks like. So each orange body is a macrophage, and you see in pink, these are all, this guy ate a couple of dead cells, this one ate one, this guy ate two. This is what you would normally see in culture and inside of a mouse, for instance, or if you were to take a human tissue, you would see these cells eating maybe one or two. But we've been able to identify pathways and perturb those pathways, and actually, using drugs or antibodies or genetic manipulations, we can actually make these cells eat many more. So here you see this macrophage eating 10 plus targets, and so they're eating many, many more of these cells on a per cell basis. So these are the sorts of exciting things that we can do once we understand the biology and perturb that biology. And then the second way that we really are trying to get at this process is changing the way these phagocytes eat dead cells. All right. So here again, this is another pathway we've identified. So a lot of these processes rely on changes in the cell's metabolism, how they use energy and how they digest the cell they've eaten. So in this video, what you're seeing here is in pink, again, you have a cell that's been eaten, and then the whole image, the whole video, you'll see there's an outline, a grayscale outline of a macrophage. In blue, you see this is labeling those parts of the cell that help degrade this eaten corpse. And in red, actually, these dots in what you're seeing is breakdown of those compartments. And so what I'm showing you here is a normal clearance process that's been perturbed. And so I want you to watch and keep an eye on these cells. You'll see this whole cell once the cluster goes away. So here you have, okay, so he's brought it in, he's digesting it. But watch what happens, and this is really exciting for us. So you see he's kind of freaking out a little bit. There's some shriveling, some shaking, and then if you watch really carefully, all of a sudden you will see, boom, the cell explodes. And so from the inside where the cell ate to the outside, you see what we call a rupture or a lysis event. So essentially, we have now perturbed the normal clearance process and made it in a way that's now unhealthy or dangerous to this cell and presumably its neighboring cells. So we'll just go ahead and watch it again, and you'll see it'll literally explode from the inside out just by perturbing one single pathway. And so these are the sorts of things we're trying to understand and look at. And so then you would ask yourself, okay, what are the consequences of such things? And so we can go to in vivo mouse models. So again, on the left, I'm going to first show you what this looks like when you target that, or when you what it normally looks like in mice. And on the right, I'll show you a three-dimensional image that is looking at targeting that pathway. And so what you're seeing here is in green, you have accessory cells of a tissue. In this case, this is in the brain. And what you have in red are what are called microglia. So these are important macrophages and phagocytes. And so if you look, this is what this would normally look like. These are all phagocytic cells. You can see in yellow, many of these cells are currently eating dead cells in vivo. And you see this, this is what it should look like. These cells, they have beautiful processes sticking out every direction. In green, you have these astrocytes that are intertwined with them. So this is happy, this is healthy, this is normal, okay? So now we've gone back and perturbed this, this metabolic pathway. And so this is what happens. So now this is the perturbed brain. And so what you're seeing here is already you can see there's a lot more red. The cells are a lot fatter. There's a lot more eating. So they're eating a lot more. They're really cranky. We call this hyperreactivity or hyperinflammatory phagocytosis. And so this is a genetic perturbation, right? So this doesn't often happen in nature. But we can do these genetic manipulations to induce this phenomenon and see really beautifully that if there's people who lack this, that's problematic. So then you're probably asking, okay, so how would you co-opt this pathway to improve cancer treatment? Well, that's exactly what we wanted to try and do. So instead of using genetic knockouts, we use small molecules or antibodies, things that can't cross, for instance, the blood-brain barrier, but can still get into the tumors in the periphery. And so what this would look like is, so we use many different mouse models, genetically driven mouse models or transplantation. We induce these models, and then we begin treatment at defined time points. And so excitingly, for instance, targeting this pathway, if we begin treatment after the tumor has already been present, we're able to have a dramatic, not just a prevention of the development of these tumors or the progression of these tumors, but we actually see full regression of, in this case, triple-negative breast cancer, which is an extremely aggressive, dangerous cancer that metastasizes, commonly metastasizes as well. And so just in this case, from working from really basic fundamental biology all the way out, we were able to identify a pathway that we could target with small molecules or with antibodies and achieve this clinical effect, at least in this preclinical model. And so this is the sort of work coming from the basic wet lab side and moving it forward, and then getting, working with our amazing physician scientists who can actually implement these treatments in their own hands as well. With that, I'd just like to thank my group. You see here, classic Korean barbecue in New York City, and I'd be happy to take any questions.
Okay, Justin, you have a lot of great questions coming in. I'm going to do my best here to summarize the major themes. So first, can macrophages eliminate cancer, or do they help cancers grow, or both?
Yeah, that's a great question. So I think Karuna highlighted beautifully that a lot of these cancers are different. And arguably that's why we haven't cured cancer, is because every single one is different. There are many cases where a macrophage's normal process, normal clearance of dead cells, helps facilitate development and progression of cancer. And the reason that happens is because when a macrophage eats a dead cell, it secretes factors that are pro-tumorigenic, that are pro these processes such as angiogenesis, such as growth factors that really facilitate it. Having said that, if you change the identity or the behavior of a macrophage, you can turn them into a cancer-killing macrophage. And this is absolutely an exciting way, can we really change those cells to do something that targets the tumors instead of helps the tumors? Yeah.
Okay, so this question's come up at least 4 or 5 times. Can they ever eat live cells by mistake?
Yeah. So this is probably one of the most important findings in our field currently, and it is actually one of the active targets of cancer immunotherapy currently. So we have evolved ways, specific proteins that prevent live cells from being eaten. And so there's a receptor on the phagocyte, and there's a receptor on the live cell that is removed or what we call downregulated when the cell dies. And these interactions specifically tell, we call these don't eat me signals. And this is extremely important because if you inhibit those signals, this is another way in which you can make a macrophage eat a live cancer cell. And so, yeah, I think that's yet another way in which we can try and change the behavior of these macrophages, that they've evolved those sorts of mechanisms. Yeah, great question.
So you hit the nail on the head with the next question I was going to ask, which was, could you tag a living tumor cell as dead in order for it to cause it to be eaten, which is related to what you just said?
Yeah, so there's several ways that we're trying to do that. One of which is get rid of or shield those proteins that say, don't eat me, I'm a good guy, don't do that. There's also other mechanisms that cells eat other cells. So unfortunately, my colleague Mike, who's giving the questions, isn't also giving a talk because he studies another mechanism by which a cell eats another cell. And so if you could exploit these other mechanisms or drive them to occur, this is yet another way in which you could have live cancer cells be eaten as well. I think obviously the concern is that you don't want also your healthy normal cells to be eaten as well. And so that's really the balance that we're working on as a field, is to try and make it so you only eat the cancer cells, which are very similar to your normal cells, right? And to do that.
We also have many questions from researchers who'd be great candidates to join your lab. I'll give you one of these. What do phagocytes do with the dead cell after processing or digesting it?
Oh, that's a great question. I love this question. Yeah, you're more than welcome in my lab with that one. So there is vigorous debate, we'll put it that way, in our field as to what happens. And in fact, any way in which you eat cells, you have to handle the stuff that you've eaten, right? The lipids and proteins and DNA and everything that's in a cell, you have to digest, you have to deal with. And so I would actually highly recommend my boss and our president, Craig Thompson's, his lab actually just published a paper suggesting some ways in which cells that eat other cells and other stuff use those nutrients in a way that is beneficial either to a host cell, but in this case, cancers can also do this, use those nutrients, those macronutrients, to enforce their own biology and help them grow. And so there's some things that are broken down and recycled and used. There are other things that we think are eliminated. So you take it in, you break it down, and then you give it back out. But yeah, this is one of those exciting areas, and I would say probably the main focus for my lab. So I'm absolutely love to host someone for that one.
Do cancer cells stop dying, or can they still die?
Yeah. So there were, I'd say a lot of the seminal work suggested that one of the things that makes a cancer is its resistance to cell death. So the resistance to the pathways that drive a cell to die. However, I would say that I think we're learning more and more that what cancer really is a competition between cells. So you might have two cancer cells competing for the same nutrient, and one wins and one loses. And the one loses might still die, whereas the one that wins had particular proteins, particular mutations, particular advantages that allowed it to live and survive, making it harder or more resistant to cell death and harder to treat later. So I'm of the belief that, and I think there's some evidence to suggest this, that at least early on during development, there's abundant cell death in the tumors. And in fact, clinical observations in the model we work with, the triple negative breast cancer, the most progressive breast cancers also have extremely high cell death in them. So that is kind of an area that we're trying to figure out is what do those dead cells contribute to, and why.
Okay, we have time for one more. So among the many great questions that are left, I'll choose one that connects the first two talks. Could you use phagocytosis to remove dormant cells, as we heard about from Dr. Ganesh?
Yeah, this is a fantastic idea. And there's dormancy, and then there's another process that happens in cancer called senescence. And so, and in fact, another one of the major scientists here at MSK who's making seminal contributions is Scott Lowe. And I would highly recommend looking up his work as well. And they recently published some work beautifully showing that you can, in fact, target those senescent cells. And so I think that's a great idea. I think you could probably identify ways in which to specifically target and clear those cells that are detrimental. And so I think, it's just a matter of identifying the targets like LICAM1 and really trying to figure out how do we get those cells instead of the neighboring healthy tissue. But I think that's a great idea. If you want to come try it, I'm happy to host you, and let's do it.
Great. Justin, thank you.
Thank you.
Well, thank you, Justin. Just to put that into some perspective, we're going to spend an hour and a half at this symposium today. The process that Justin just described to you will take place in each of us over that hour and a half 10 billion times. So this is a really important normal process, and we understand from his answers and from his presentation that it affects cancer in many ways. In fact, the question that was asked that Michael was so happy to ask, which was, can cells ever eat normal or living cells, was actually discovered by our dean, Michael Overholzer, 10 years ago, and led to some of the discoveries that Justin summarized. And so we're very proud that multiple people work on this. And then finally, we don't just study sometimes cancer here. Another group here that worked with Justin has discovered that when you don't make normal macrophages that can engage in this process of clearing dead cells, it's the last remaining genetic cause, we believe, of cardiovascular disease, and leads to increased risk associated with certain forms of cardiovascular disease of the inability to clear viruses such as COVID-19. So all of this comes full circle. By understanding the basic biology that cells engage in and what can go wrong, we learn not only about cancer, but about other diseases such as cardiovascular disease or infectious disease. So with that, I want to now turn to our final speaker, who is Thomas Norman. Thomas is an assistant member in the Sloan Kettering Institute's Computational and Systems Biology program. Thomas is originally from Canada. He earned his undergraduate degree from Queen's University in Ontario, and he got his PhD degree at Harvard. Since he's joined us, Thomas has been developing computational tools and single-cell techniques to build predictive algorithms of cancer cell behaviors. This is the exciting and evolving field of functional genomics, and I'm pleased he's going to come up here and share some of his most recent work with us. Thomas.
All right, thank you, Craig, and thanks everybody for coming to my talk. So I am Tom Norman. As you might have seen flashed up at some point, I am a systems biologist, and I am also a geneticist. So I'll start with the easy one. Geneticist means that I study genes. Systems biologist means that we look for systematic approaches to study those. So gather lots of data and look for principles behind how things behave. And so my talk today is actually going to focus on the data generating side of this equation, where it's how can we get a lot of information in order to build sort of computational models of cellular behavior. And so I've called it a million tiny experiments. And my goal here is first to sort of convince you why we might want a million experiments, why we might want them to be tiny, and then what we can do with this capability if we can realize it. So just to get us all on the same page, I thought we'll take a real zoom out. As we've just discussed in detail, the body is made up of trillions of cells of many different types. The precise number is actually a matter of debate, and a distressing number apparently eating each other. So the interesting thing about these different cell types is that actually all of them share the same genetic material, the same DNA. And what they differ is in how they actually use that material. And this means that they express different genes. And so there are about 20,000 genes in the human genome. Each cell type expresses 5 to 10,000 of those, and that's what makes effectively the difference between a liver cell, a heart cell, a brain cell, et cetera, which genes are on or off. And so this analogy that I'd like you to think about this is to think of genes almost as notes in a piece of music or keys on a piano. And that's because I'm trying to capture this idea that genes actually have to work together in order to realize their roles. Each gene has to be on at the right place and the right time in order for this cell's behavior to be correct, in the same way that you need every note to make a piece of music. And so this brings us obviously then to the question of what happens when something goes wrong. And that is a mutation, as Karuna discussed earlier. A mutation is an error in a gene that affects its function. Many of these will lead to heritable genetic diseases. I have one example up here, which is sickle cell disease. This is caused by a mutation in the gene for hemoglobin, which carries oxygen in the red blood cells in your blood. If you have this mutation, your red blood cells will sometimes adopt this characteristic sickle shape, and they will not transport oxygen as well, which leads to the disease. So in this music analogy that I've introduced, this is the equivalent of having one of these keys maybe be a little sticky or something like that. And the task of genetics is to come up with ways to find that gene that's defective somehow. And you could sort of imagine with a single gene that maybe this task is something fairly manageable. Maybe you listen to this piece of music over and over again. You can sort of detect that a piece is missing, and then maybe you can identify it, and then maybe we can target that. And that's what underlies a lot of exciting emerging therapies for genetic disorders. The unfortunate reality, though, is that this picture of a single gene being defective and driving disease, unfortunately, is the exception rather than the rule. The rule actually is so-called complex genetics or polygenic disorders, the simplest example of which is cancer, which almost always arises as the result of a combination of different mutations. And sort of to add insult to injury, the same disease can often arise in many distinct ways. And let me just sort of spell this out for you. The sort of emerging standard of care in a lot of cases for cancer treatment is if you're a patient and you go to the hospital, they'll actually take a little piece of your tumor, they'll sequence it, and they'll identify the mutations with the idea of trying to find some sort of targeted therapies that might help you. And what I'm saying is that if another patient walks through the door and we sequence their tumor, some of the mutations might be the same, but often many are sort of distinct to each individual. And so you can see then that this makes the task of genetics quite difficult because, first of all, now if I'm listening to this sort of a piece of music, first of all, I have to try to identify not one missing note, but maybe 3, which you can imagine gets more and more difficult. And then, as I said, if a second person comes in, I'm looking for a completely different set, potentially, of missing notes. And so just to spell out this sort of logic, I want to just share a little thought experiment with you, which is if we sort of limit ourselves and we say we've somehow figured out that only 200 genes potentially influence a disease, if that disease is caused by one mutation, that's 200 possibilities. If it's caused by 2 mutations, if we do the math, it's actually about 20,000 possibilities. That's the number of ways of choosing 2 things out of 200. And if it's caused by 3 mutations, it's 1.3 million. So we can see that these sort of like many possibilities, once we start considering polygenic disease, really run away on us quickly. This is called a combinatorial explosion. And so we have this sort of pressing question of how can we possibly investigate things of this scale if we want to start understanding how polygenic diseases arise. And this, of course, brings us to the title of my talk, now featuring an exclamation mark, 1,000,000 tiny experiments. And so what I'm going to talk about is our approach to gathering data on a scale that's necessary to start studying these sorts of behaviors. And I'm going to add a little proviso on the bottom here. We're going to say at least this is the goal, because this is something that we haven't actually achieved yet, though we're well along the way. And so how could we actually do 1,000,000 experiments? One way that you might think of, which is actually not an unreasonable one, is perhaps we could go find 1,000,000 patients. The problem, of course, is that this is very slow, it's very expensive, and potentially it might actually in many cases be impossible. There might just not be enough people with a given condition. And so this forces us back to the drawing board to start thinking of things that we have in abundance where we could start asking these sorts of questions. And actually, the organoid systems that Karuna mentioned are one possible candidate. I sort of went one step further to sort of the smallest possible thing that you can think of, and that is to think of using cells as experiments. I told you at the beginning that your body is full of trillions of cells, so perhaps you can spare me 1,000,000. So we're going to try to use cells as experiments in order to understand polygenic disease. And so here we, of course, benefit from what is now a certifiably seminal discovery, just awarded the Nobel Prize in Chemistry to Emmanuelle Charpentier and Jennifer Doudna, who are up here, for CRISPR genome editing, which you may have seen in sort of the popular press. And so how does CRISPR work? This is a cell, and here are our genes again. And CRISPR is actually two things. It's a protein called Cas9 together with a little bit of genetic material called a guide. And it basically functions as a kind of targeted missile where what this thing is doing is it's constantly kind of landing at different points in the genome, and it's testing whether it's able to bind by sort of comparing its guide to the sequence of the genome below. And so what I'm showing you here in this animation is it's trying to land on this yellow gene, and it doesn't match, so it sort of bounces off. What happens now if it lands on the next gene is that the guide matches, and at least in this sort of original system, what happens is that actually triggers Cas9 to cut. It introduces a little break in the DNA, and this actually inactivates the gene underneath. So this is a way of turning genes off. And so I'm not going to get into the details, but there's been a sort of explosion of technologies built around this concept where now we can turn genes off, we can turn them on, we can turn them up, we can turn them down, we can do all kinds of things. And so you really should think of CRISPR genome editing as more like a toolbox for manipulating our genetic material. And so this is how we're going to turn our cells into little experiments, because this gives us a very easy way to programmably alter different cells. So for one thing, we can take one cell and we can put in a guide that maybe turns gene A on. We can take another cell and we can put in a guide that maybe turns gene B off. And the sort of true magic of this technology is that it's actually trivial to sort of combine these effects. So in the third cell, we can actually put both together. We can turn gene A on, that's supposed to say gene B off, but we can have both effects at the same time. And so this lets us start investigating these polygenic combinations where we're altering multiple genes at a time. The next thing that we have to do is actually figure out what those changes are doing. And so we need a way to measure these cells. And so here we rely on a quite magical technique from engineering called microfluidics, which allows us to encapsulate these cells in tiny little droplets. And I'm going to show you a movie that I have taken from the University of Southampton because they have a much more beautiful thing than anything that I have. So this is looping. And what we see coming in here on the left is a cell. That's that little sphere. And then what you can see is it's almost being sort of injected into these droplets that are being created. And you can create thousands or millions of these droplets at a time. And what's important to think about is that these droplets can be completely controlled. We can control how the cells go in. We can control what goes on inside the droplet. And so they're a way of making really small experiments. And so that lets us make these experiments have sort of massive capacity. And so we can do experiments inside the droplets. So this is the sort of general thrust of how we do these experiments. We take our guides, which program the genes or combinations of genes that we want to change. We put those into cells, thousands or millions potentially of cells, and we change 1 to 3 genes per cell. Then we put those cells into droplets using the microfluidics approach. And then finally, we have to measure them somehow. And there are a lot of different ways or a lot of different things that we can now do to cells and droplets. The one that I am best known for is an approach to actually measuring gene expression. So seeing which other genes turn on and off. And that's because, remember, genes work together. So if I disrupt or if I turn on gene A, often there will be knock-on effects on genes B, C, and D. So this gives us a way to gather truly massive amounts of information. So one of our experiments might gather 500 gigabytes of data at a time. And so obviously summarizing something like that, particularly in a 15-minute talk, is quite difficult. So I will just show you a visualization of our biggest experiment so far, which is 110,000 cells. Notably, I did not call my talk 110,000 tiny experiments because it does not quite roll off the tongue. But what's going on here, now note this is not the droplets, this is a visualization. And so each dot here is a cell, and I've colored cells that have sort of similar behaviors. And what we could see is we're sort of taking this population of cells, we're pushing them in a whole bunch of different ways by sort of genetically manipulating them, and then we're seeing what happens to their behavior as a result. And there's actually kind of only a limited repertoire of things that cells can do, which is why there's kind of these clumps of behavior. Now, I could sit here and talk about this for another 15 minutes, but we'll save that for next year. So I just want to close with this idea then of why a million tiny experiments. And I've sort of already set this idea up, and it's that there are actually many diseases where it's difficult to get enough patients to study them using traditional population genetics approach. And so what we're presenting here and what we're developing is not really an approach to any particular disease, but rather one that can be sort of broadly applied to many diseases where we would like to find out what's going on. And so I just want to give you 2 very quick examples of things that we've looked at. One is an application in a cancer called CML. And here what I'm just showing you is one of the combinations that we found, which is just a combination of genes where what it does is it actually takes the cells and it turns them from being a cancer back into being red blood cells, essentially. This is called a differentiation therapy, and this is why this pellet of cells that I'm visualizing here turns from a sort of whitish color into a red one. The second application, just to emphasize something very different, is something that we just started doing that I'm very excited about, which is in rheumatoid arthritis, so not cancer. This is collaborative work with people from across the street at the hospital for special surgery. This is some cells out of someone's knee. This is the kind of thing that a scientist gets excited about. And what we're seeing here in this fluorescent micrograph is just that the same machinery that we used before has been delivered to these knee cells. And so we have reason to expect that we should be able to do these experiments. And so lots of things to do. And so I just want to emphasize for this audience, a lot of people are probably thinking about where they might go to college. I just want to emphasize our work is very interdisciplinary, and sciences in general. We have things that we're trying to do in sort of engineering. We have things that we're trying to do in computer science. We have things that we're trying to do in biology and medicine. And the composition of my lab is actually made up of people who come from these different backgrounds. And so there's a lot of space in modern life sciences research for different approaches. And so with that, I will leave you with my parting message, which is just, I think, this core idea that our genes work together to influence our health and disease, and that we can start using these sort of massive experiments to understand these effects. So with that, I will take any questions.
Okay, Tom, great. So we'll start it off with this one. You mentioned that most cells express 5,000 or 10,000 genes. Do some cancer cells express more genes or fewer genes?
Yes, in general, cancer cells express more genes than so-called primary cells, which are ones that are taken out of your body that are sort of in a normal state. And in particular, actually, there's a huge variation across different cell types of how many genes are expressed. So things like neurons don't express very many genes at all. I actually don't know the answer to the do some cancers express fewer genes question, so I guess I've lost the stumping question.
Okay.
Yeah.
Fantastic.
Yeah.
How do you rule out genes to get to the possibilities down to 200? A few questions about why you landed on 200.
Yeah, so that's actually something that's informed by how science is actually done, which is that, I'm up here talking about what we call systematic or unbiased approaches, where maybe you're sort of trying to test all of the possibilities. In reality, often people have worked in some system for years or decades, and they actually know a lot about it. And there are sort of other approaches that they've used to implicate genes that might play a role in that process at hand. So you kind of can get a sense of what things are close to what you're interested in versus things that are very unlikely to play a role.
We have several questions about the different cell behaviors that you're measuring, and also how do you actually measure gene expression? I wonder if you can answer maybe both of those.
Yeah, so that actually is figure is actually from a paper that we published. The main thing that's going on there actually is changes in cell fate. We're actually monitoring differentiation. And so what we see in a lot of things is particularly when we turn new genes on, what you actually do is cells will start to convert from one cell type into another. This is partly a feature of cancer cells because they tend to be more plastic, is the word, in terms of their phenotype. And so often you can find ways to push them into sort of quite distinct new states.
Okay, 1 second. So how can tumors with different genetic mutations act in the same way?
Oh, well, that is, I think, an unresolved matter. This is actually sort of the central hypothesis behind so-called systems biology, which is that perhaps there's some sort of greater organizing principle that we as scientists don't understand yet, where if we look at at a different resolution, say, than at the level of genes, then maybe there are states that we can understand better that are shared.
Okay. Can you work backwards? Can you find combinations of genetic mutations and see if they create the disease?
Yes. So we've actually considered doing both directions. And there is no, because, you can take either diseased cells and work backwards to try to sort of rebuild a healthy state, or you can take healthy cells and introduce disease-causing mutations. This is actually particularly germane to cancer, where there are many known causal mutations, and actually often the question is more, what are those things doing together, rather than what they are.
What are some of the limitations you have come across with cell experiments?
Okay. So, well, I could think of one very simple one is cost. So I'm hiding the fact here that these experiments are very expensive, though they will go down in cost, and that's one of the things that we spend a lot of time doing. The other thing is that these approaches are what we call in vitro, which means that they're happening in sort of idealized circumstances that may not accurately reflect the conditions in the body and things like that. And that's actually why it's important for people like us to work with people like Karuna, who can do things like build organoid models or work in mice that might be more reflective of conditions in the body. We're more like a hypothesis generating machine.
You have numerous questions about genetic interactions.
Oh,
so I'll read one. Could you look at the way that one cell with mutations in genes A and B would grow in a heterogeneous population?
Wait, sorry, say that again.
Basically, if you have mutations in a gene A and a gene B in one cell in a mixture, how do you look at that interaction between genes?
Yeah, so this is, this was actually my main work before coming to MSK. And so You can actually sort of, what you do, like I said, you can think of genes as ways of pushing cells in different directions. So if I change gene A, I'm pushing it in one direction. If I change gene B, I'm pushing it in another direction. And the thing is that sometimes when you put A and B together, it actually goes in a third direction. And so the way that we sort of parse that out is that we have these computational models that let us break the sort of effects that we observe in those doubly perturbed cells into effects explained by each of the single genes plus things that we don't understand. And obviously, if something completely new is happening, then that third category ends up being full of information, essentially.
Great. Okay, Tom, we have time for one more. Okay. Can CRISPR-Cas9 be theoretically used to deliver a sort of genetic package to induce the death of cancer cells and not others?
Yes, so this is actually one of the things that we're most interested in doing. This is a concept called synthetic lethality, which is that often cancers, because they have different biology than normal cells, have sort of different sensitivities. And so you can hopefully find a gene that you can target that maybe is more important for the cancer cells than it is for normal cells. And then if you develop a therapy that targets that gene, it's selectively lethal to the cancer while leaving the normal cells happy.
Great. Tom, thank you very much.
Thank you.
Well, those were fantastic questions, and I want to thank everyone who has attended to make this such a successful virtual event. Nearly 1,000 of you have signed up to give you some scope for those that have been listening. You represent nearly every state in the United States and 34 different countries. I remind you, we are available. There are updates on all of the work that you've heard today on our social media posts from Memorial Sloan Kettering that comes out on a daily basis. And also you can visit the lab websites of the 3 individuals that you've seen here today. You'll also be receiving follow-up emails that will have information on future major trends events if you're interested in seeing more of this programming, and in some of our summer programming that offers internships and other programs to get to know our research enterprise even further and even give opportunities to participate. So with that, I want to thank all 3 of the speakers for tremendous presentations. Mike for being the interpreter of the many questions that were asked so that we could get them all in a meaningful time, and all of you who attended this very important and first virtual event in our Major Trends series. Thank you all.